Hello everyone, welcome to this amazing platform of Infinity Learn by Sri Chaitanya. My name is Kiran Chaudhary and I am your biology faculty. Students, let's talk about today's session. Today we will discuss about replication, machinery and enzyme. Let me remind you what we did in the last class. In the last session, we discussed about the introduction of the replication process as well as the scheme suggested for replication of DNA by Watson and Crick, and we also took the sneak peek of how exactly the experimental proof was given for semi-conservative mode of DNA replication that we covered in the previous section. In today's session, we will try to understand how DNA replication takes place, first thing, and second, what are the various enzymes that actually helps in executing or completing this DNA replication process. Hello everyone, hello Shashank, hello Sunil, good afternoon, very warm welcome in today's session, right? Let me know wherever you are not able to follow, okay? So put your doubts in the chat section. Whenever I will find time, I will definitely come and check that, okay? So let us start today's session. Before that, I would like to tell everyone that I request you all to visit infinitylearn.com website and check out the courses that we offer there. You can avail or use those courses at 40% off by using the code KIRAN40, K-I-R-A-N, K-I-R-A-N 40, KIRAN40 code. Apply this code to get 40% off on all the courses and subscriptions that we offer at infinitylearn.com website. Let us start now. So see students, DNA replication mechanism. The replication mechanism requires a set of enzymes because you know it is a process that is taking place where it is a process that is taking place inside a cell, inside a living system, and obviously for processes to take place or reactions to take place inside a living system like cell, there is requirement of enzymes, and obviously here also for replication process there will be requirement of enzymes, not only one or two but several sets of enzymes, right? So there is requirement of a set of enzymes. There will be several enzymes that will participate in this process. Talking about the most important one, the most important is DNA polymerase. Remember, the name itself is DNA dependent DNA polymerase. I repeat myself students, the main enzyme is DNA dependent DNA polymerase. Let us try to understand why such name. The main enzyme for replication process is DNA dependent DNA polymerase. Why the name so? Because this enzyme will use the parental DNA molecule as template to catalyze the polymerization of deoxyribonucleotides, right? See here, what is polymerase? If I talk about the polymerase is an activity where this particular enzyme will do what? Polymerize the deoxyribonucleotides, right students? So polymerase का क्या मतलब है? The polymerase activity means to polymerize the deoxyribonucleotides, means connecting one deoxyribonucleotide to another, to another, to another. So like this way, one will be connected to another, one will be connected to another. So इस तरीके से catalyze करेगा ये एक deoxyribonucleotide को दूसरे से तीसरे, like this polymerization of deoxyribonucleotides will take place with the help of this enzyme. Second, why it is known as DNA dependent? Because it will utilize the parental DNA molecule as a template for catalyzing this polymerization process, right? I hope this much is clear to all of you. This enzyme, let us talk about some special characteristics that this enzyme possesses. First, it is highly efficient, right? Highly efficient, I can give you an example for, see, if we consider an organism known as E. coli. This example is given in your NCERT as well. If we talk about or consider E. coli or replication process that take place inside E. coli, replication process of E. coli gets completed in 18 minutes, right? And you know how many nucleotides or how many base pairs rather I should say are present in the DNA of E. coli? So, E. coli DNA possess 4.6 into 10 raised to the power 6 base pairs, and the polymerization of such high number of base pairs, 4.6 into 10 raised to power 6 base pair gets completed in just 18 minutes. Isn't it amazing? And this shows the power, the efficiency of this enzyme known as DNA dependent DNA polymerase. How efficient it is. Next, it possesses high degree of accuracy. Yes students, accuracy is very much required because even a single error in DNA during DNA replication process can lead to undesirable mutations, right? So, these two properties are very much required in a DNA dependent DNA polymerase. Let us see next. See students, the entire replication process we will study under these 5 steps. The entire DNA replication process we will study or understand with the help of these 5 steps. We will start with the first one, origin of replication, right. So, remember we will take each and every step and I will try to explain you from starting to end. So, before like starting means starting from where exactly the DNA replication process will start, and ending means where you will get or started getting the polymerized strands of daughter DNA, right. So, this is how it is going to move. So, we will start with the very first step, that is origin of replication. See students, what is origin of replication? Origin of replication, DNA polymerization, right. This replication process of DNA does not start anywhere on the entire length of the DNA. Rather, this DNA polymerization will start with or at a particular point on DNA known as ORI. So, DNA replication starts at a particular region of DNA known as ORI, right. So, these are the specific places which are capable of starting DNA polymerization, right. Next, second thing that you need to understand is the activation of deoxyribonucleotide. ध्यान से सुनना स्टूडेंट्स, क्या होगा, अब जितने भी deoxyribonucleotides हैं वो सारे के सारे deoxyribonucleotides will try to get converted into deoxyribonucleoside triphosphates, right. See how. DAMP, DGMP, DCMP and DTMP. What are these students? These all are monophosphates. These all are monophosphates, right. Now, all these types of monophosphates will react with 2 molecules of H3PO4. What is H3PO4? It will be a source that will provide inorganic phosphates. In the presence of phosphorylase enzyme and also will involve energy. Energy need to be given in this reaction. See, so each and every type of deoxyribonucleotide, or rather I can say deoxyribonucleoside monophosphate possessing single phosphate, each one will participate, each one will react with 2 molecules of H3PO4. As a result, what will happen? The 2 phosphate group, inorganic phosphate group will get attached to this monophosphate, right, deoxyribonucleoside monophosphate, it will get attached to it, and as a result what you will get? As a result you will get a deoxyribonucleoside triphosphate. Remember, this reaction will be catalyzed with the help of phosphorylase and there will be requirement of energy in it. So, finally what you will get DAMT, sorry, 1 second, I wanted to write T here. Because here M is representing to what? Here N is representing to monophosphates. So, DATP and DGTP, DCTP and DTTP. So, what are these? These all are triphosphates. So, conversion of monophosphates into triphosphate. This is known as activation of deoxyribonucleotides, monophosphate ones. So, conversion of deoxyribonucleoside monophosphate to triphosphate and this is how it will be executed. अभी तक हमने क्या पढ़ लिया students? ध्यान, try to recall what are we doing? We are trying to understand how replication of DNA will take place. That's the objective of today's class. हमें ये समझना है कि DNA replication कैसे होता है. DNA replication का प्रोसेस अपने आप में कॉम्प्लेक्स प्रोसेस है. एक साथ हम लोग सब कुछ नहीं समझ सकते. तो पूरा का पूरा प्रोसेस मैं आपको 5 स्टेप्स में समझाऊंगी. First we will discuss the entire process into 5 steps. So I have divided the entire mechanism of DNA replication into 5 steps so that it becomes easy for you to understand. उसके बाद after explaining the 5 steps, I will directly with the help of an image, I will try to explain the entire process in one shot or in one go. But for that, to understand that whole thing in one go, you need to understand the whole process in step. So that is why we are doing this step-wise understanding. और अब तक हमने 2 स्टेप पढ़ लिए हैं. सबसे पहला स्टेप क्या था? ORI. Right? So, DNA replication will not start at any random place on the DNA, rather it will start on a specific place on the DNA molecule known as ORI. And second क्या था? Second था activation of deoxyribonucleotides जो कि इस तरीके से हो रहा है. अब important ये है कि all should think or this question should strike to everybody's mind that why this activation is taking place. What is the requirement of activating deoxyribonucleotides? It is a good question. Now let us try to understand why this happens. So activation of deoxyribonucleotide is required for 2 things. Deoxyribonucleoside triphosphate serve as a dual purpose, act as a substrate as well as provide energy for polymerization. So see students, you know what are we studying? We are trying to understand DNA replication and DNA is made up of what? It is made up of multiple or it is a chain of deoxyribonucleotides, right? So obviously the form deoxyribonucleoside triphosphate first of all will act as a substrate for polymerizing the deoxyribonucleotides, right? Because of these only the DNA molecules, the daughter DNA molecules will be made up of, right? So first thing is substrate. Substrate for which enzyme? DNA dependent DNA polymerase, right? Second thing, they will provide energy for polymerization. See students, replication is energy wise very expensive process. Replication is energetically very expensive process. And when I say replication is expensive process, that simply means lot of energy will be involved during replication. So these deoxyribonucleoside triphosphate will provide energy. Yes. So see students, try to understand this very simple representation. This is what I am trying to make. I am trying to make, what is this? This is a monophosphate from here to here, monophosphate. Which monophosphate is this? This is deoxyribonucleoside monophosphate. Deoxyribonucleoside monophosphate. Now, what you will do is, during the activation of deoxyribonucleotides, what you were doing? You were connecting or binding 2 more additional inorganic phosphate with this molecule, right? So, this. So, see students, these additional 2 inorganic phosphate will actually have what? Lot of energy in their bonds, right? So, during the replication process, during the polymerization of deoxyribonucleotides, in this replication process, what will happen? These terminal phosphate, these two terminal phosphates will break, and because of the breaking of the bonds of these 2 terminal phosphate, there will be release of energy, and released energy will be utilized to polymerize the deoxyribonucleotides during replication process. That's the dual purpose of deoxyribonucleotides, right? I hope this much is clear to all of you. So, you have formed this, see. In this process, what we did? We exactly did this. So, this was available DAMP. So, this is an example. So, this is an example of DAMP. So, this much was an example of DAMP, and after this phosphorylase activity and addition of 2 phosphate with the help of H3PO4, finally what you converted? Finally, you converted this DAMP to DATP, right? So, this is what? DATP, right? Triphosphate, deoxyribonucleotide triphosphate, right? So, in this, these 2 terminal phosphate possess high energy. So, when this 2 terminal phosphate will break, released energy will be utilized in this process, and the remaining DAMP molecule, after breaking of these 2 terminal phosphates, after breaking the bonds of these 2 terminal phosphate, the remaining DAMP molecule can be utilized as a substrate for polymerizing, right? I hope this much makes sense for everyone. चलिए आगे देखते हैं, आगे हमें क्या करना है, नेक्स्ट स्टेप में हमें क्या समझना है. अभी तक हमने क्या किया, हमने अभी तक तैयारी की. नाउ लेट अस स्टार्ट द प्रोसेस. In order to start replication, the parental DNA molecule needs to unwind, right? So, this is, for example, this is the parental DNA molecule. This is the parental DNA molecule. During replication, what will happen? During replication, unwinding. Unwinding means opening. Unwinding means opening of the DNA molecule or separation of the 2 strands in a DNA molecule. So, like this, okay. But remember, who is going to catalyze this process? The enzyme known as helicase. So, unwinding or unwinding will be catalyzed with the help of the enzyme known as helicase. So, enzyme helicase will actually help to open up or separate the 2 strands of the DNA, parental DNA molecule participating in DNA replication process, right. So, unwinding will be catalyzed with the help of enzyme helicase. Next is the exposed or separated out strands of the parental DNA molecule need to be stabilized. Otherwise, what will happen? You know, both these strands are what? They are complementary to each other. So, there is a tendency that even after opening, they will again come closer and form hydrogen bonds between them. And if this will happen, then how the replication process will start, right? In order to start replication process, right, in order to proceed the DNA replication process further, the two strands need to be stabilized in separated form. To stabilize these two separated DNA strands, there is requirement of single strand binding protein. This protein will help to stabilize these two strands in a separated form till the replication process starts and the daughter DNA strands started getting synthesized, right. So, who is going to help? Second, who is going to help? Single strand binding protein is going to help out. Now, one more thing you can obviously see and observe here, students. What you can see and observe is when this replication, right. So, when helicase is acting in trying to separate out the two strands of the parental DNA molecule participating in replication process, see here, the separated out strands of the DNA molecule look like Y or forms a replication fork that has actually Y shape or possess a Y shape. So, formation of replication fork which is actually Y shape. So, when does this replication fork get formed? I repeat myself, students, ध्यान से सुनिएगा, ये replication fork कब बनता है? जैसे ही हेलिकेज अपनी एक्टिविटी शो करना स्टार्ट करता है. As soon as helicase starts showing its activity by separating the two strands of the DNA molecule, hence because of the helicase activity there is separation of the two strands, and as a result the separation of the two strands makes a replication fork which looks like Y in shape. Second, the separated strands need to be stabilized so that replication process can start and proceed on those strands. What is this? This is SSBP, SSBP, single strand binding protein. Single strand binding protein, right. So, why single strand? Because it is trying to stabilize, it is trying to stabilize the separated out strand, right. I hope this makes sense. Next is supercoiling. Yes, students. So, what happens? What happens? So, see, if you see this, if you observe this diagram that I tried to make for you, you can see this. Now, replication will move like this. This is the direction in which replication will further proceed, right. And this is what? This is your replication fork. This is what? This is your replication fork. Replication fork. So, just opposite to the replication fork, there will be supercoiling in the DNA molecule because of the activity of helicase. Helicase is trying to separate out the two strands, right. And just opposite to the replication fork, what will happen? Because of this opening of the two strands of the DNA molecule, there will be creation of tension in the DNA strand. So, the segment or part of the DNA which is opposite to the replication fork will start getting supercoiled. Because of the development of tension in the DNA molecule, there will be supercoiling seen in this portion of the DNA, and hence there will be requirement of another enzyme known as topoisomerase. What this topoisomerase will do? It will try to release this tension so that supercoiling can be prevented, and this helicase can act smoothly and DNA replication can be further continued without hindrance, right. So, supercoiling is released, the tension that is developed because of the supercoiling will be released with the help of enzyme known as topoisomerase, and similar activity in prokaryotes will be done by DNA gyrase. Next, students, next that you need to understand over here is formation of primer strand. What is a primer strand and why a primer strand is needed? These are two important questions. See students, DNA dependent DNA polymerase. You understand, this is the main enzyme. DNA dependent DNA polymerase is the main enzyme, DNA dependent DNA polymerase main enzyme for this replication process. But this enzyme has one incapability, right? It cannot start the replication process. It cannot put that first nucleotide in order to start the synthesis of DNA or in order to start the replication process. It cannot do that. So, it requires somebody to put that nucleotides or first nucleotide so that it can continue the process of replication or polymerizing the deoxyribonucleotides. So, exactly here comes the importance of the primer strand. So, primer strand actually helps DNA dependent DNA polymerase to start the replication process, right. So, DNA polymerase is incapable of initiating DNA synthesis or DNA replication process. Primase is the enzyme that will help to synthesize a short primer. Not to forget, what is primer? Primer is a strand of RNA, a short strand of RNA synthesized by primase. Primer will start the errorless replication. What is the function of the primer? To start the replication process errorlessly. Next, primer will be removed enzymatically on completion of initiation of DNA replication. So, once the initiation of DNA replication is perfectly completed, there won't be any requirement of this primer and primer will be removed because primers are not required, they are RNA and RNA is not required in the DNA molecule, right. So, I hope everybody should be clear why primer is required, right. Why primer is required and what is a primer. I really hope this much is clear to all of you. Next is, now till now we have reached to a point where we have started the replication process. So, starting of the replication process is clear to all of you. Now, let us see how elongation is done. So, elongation of the new strands will be done obviously with the help of DNA dependent DNA polymerase. This enzyme was actually incapable to start, but once this DNA replication is initiated properly with the help of RNA primer, now the whole load comes to this DNA dependent DNA polymerase where it can very efficiently polymerize the entire strand. See here, very important thing that you need to understand is DNA dependent DNA polymerase can polymerize the deoxyribonucleotides, polymerize the deoxyribonucleotides in one direction only, 5:3. This is the limitation of DNA dependent DNA polymerase. It can show its catalytic activity only in the direction of 5:3. It can form bonds or it can polymerize, it can connect the deoxyribonucleotides only in this direction. And because of this during replication process, this is what, this is a parental DNA, this is the parental DNA. And remember one very important fact students, during DNA replication both the strands of the parental DNA will act as a template. This is not the case when I will teach you about transcription. So, this is very important difference between transcription and DNA replication. So, during DNA replication both the strands of the parental DNA molecule will act as a template. That is why here parental strands, right. So, both these strands will act as a template. Remember this, what is template? Template is the strand on which new strand will be synthesized with the help of DNA dependent DNA polymerase. Remember both will act as a template, both the strands of the parental DNA. But because of the limitation that DNA dependent DNA polymerase can show its activity only in the direction 5:3, because of this there will be a continuous synthesis. There will be a continuous synthesis only on the parental strand which has the polarity 3:5. So, the parental strand that has polarity 3:5. I repeat myself, students, the parental DNA molecule, the strand that possesses the polarity 3:5, on this template strand there will be continuous synthesis of the strand, and the strand of the parental DNA molecule that possesses the polarity 5:3, on this the newly synthesized strand will be discontinuously formed. So, there will be a discontinuous synthesis of the new strand on 5:3 strand of the parental DNA molecule. And why this happened? Because DNA dependent DNA polymerase can show its activity only in 5:3 direction. That's the only reason because of which this newly synthesized strand, if I talk about, on one template they get synthesized continuously and on another they get synthesized discontinuously, right? Please remember this. So, in your MCQ, even in your exam, NEET exam, please do not get confused. This is a very important point, that on which strand of the parental DNA molecule there will be continuous synthesis and there will be discontinuous synthesis. So, replication is continuous on 3:5 strand of parental DNA, right, and that formed strand, this continuously synthesized strand, is known as leading strand. It is known as leading strand, and this discontinuously. So, replication is discontinuous on 5:3 strand of parental DNA and formed this strand will be known as lagging strand, right, lagging strand, and the fragments will be known as Okazaki fragments that will be connected or joined later with the help of DNA ligase. Right, students, this much is clear? Perfect. So, that's the whole thing that I wanted to explain in today's session, students. Now, let us try to take a small recap of the entire process of DNA replication. Remember, I told you I will explain you in 5 steps, right, because it is a complex process. I cannot explain you everything in one go because that will make the understanding complex. So, we understood the whole process in 5 steps. Now, let us take a quick recap of the entire DNA replication process. Remember where it will start, not random places, only at the specific place known as ORI, where origin of replication, ORI, original of replication, only at those specific places replication process will start. Secondly, see this, what is this? DNA polymerase. This is RNA primer. This is DNA ligase, right? Now, remember what is this? This is how the replication process looks like, and let us try to understand each and every element's activity. So, we will start with this. What is this? This is helicase. Remember, this replication process is starting at ORI, where what helicase will do? Helicase will try to separate out the 2 strands of the parental DNA molecule, where these separated out DNA strands will be stabilized with the help of single strand binding proteins. They are trying to stabilize the separated out strands, right, and because of this helicase unwinding the DNA molecules, there will be supercoiling in the DNA opposite to the replication fork, which will be released with the help of topoisomerase. Topoisomerase will release the tension in this part of the DNA, right. Secondly, what you need to understand is there will be requirement of an RNA primer synthesized by primase enzyme, which will help to initiate the DNA replication mechanism. Once the initiation of DNA replication mechanism is properly completed, then who comes into the play? DNA dependent DNA polymerase that will do the elongation process. And remember, if you notice this very nicely, this strand is what? This is the 5:3 strand of the parental DNA, on which on which lagging strand is getting synthesized, and this is the 3' is the 5' strand of the parental DNA on which leading strand is getting synthesized, and these are the Okazaki fragments that will be joined with the help of DNA ligase later, right? I hope this much is clear to all of you students. That's all from today's session I wanted to explain. Alright, I hope this whole thing is clear to all of you. Let me know if there is any doubt till now. I hope, Sunil, your doubt is completely resolved now, right? Okay student, that's all. Thank you for joining my session. Give me a thumbs up if you like me and this session, and do not forget to subscribe Infinity Learn by Sri Chaitanya. Also, do not forget to hit the bell icon so that you get notified every time whenever I'm going to come live. That's all. Thank you. Bye-bye.